Welcome back students. A molecular basis of inheritance unit la we will continue with the next video. So in this video we will study about the packaging of DNA. Packaging of DNA. So packaging of DNA vandute can be also called as the condensation of DNA. ஏன்னா நம்ம இப்போ டிஎன்ஏன்னு ஒரு ஸ்ட்ரக்சர் படித்தோம் இல்லையா, அது லென்த்தி த்ரெட் லைக் ஸ்ட்ரக்சர். அந்த கம்ப்ளீட் ஸ்ட்ரக்சர் வந்துட்டு 23 pairs of such structures, very long structures வந்துட்டு has to be packed inside the nucleus of a microscopic cell. செல் மைக்ரோஸ்கோபிக், அந்த செல்குள்ளே இருக்கிற நியூக்ளியஸில் வந்துட்டு 23 such pairs has to be packed in all cells of the body. அப்போது அந்த லென்த்தி டிஎன்ஏவை வந்துட்டு எப்படி வந்துட்டு செல் மேனேஜ் பண்ணி அந்த நியூக்ளியஸ் அப்படிங்கிற ஒரு சின்ன ஸ்ட்ரக்சருக்குள்ளே வைக்கிது அப்படிங்கிறது தான் வந்துட்டு தி condensation or packaging of DNA. This is by formation of a structure called nucleosome. So nucleosome அப்படினா என்ன அப்படினா, it is going to be the structure that helps in the packaging of DNA. So this nucleosome is also called as, இது நியூக்ளியோசோமோட இன்னொரு நேம் நான் இங்கே எழுதுறது, they are also called as V8 bodies or new bodies. So the DNA with nucleosome formation is converted into a structure which we call it as chromatin fiber or simply chromatin. So DNA with nucleosome formation vandittu will be called as a chromatin. Chromatin vandittu is defined as beads on string structure. So a string containing beads, that is what is called as beads on string structure. So DNA with that is the chromatin is defined as bead on string structure, and this such a structure was found inside by a person called Woodcock. Woodcock, he said that these chromatin fibers, before the identification of what is a nucleosome itself, Woodcock vandittu when he was studying about DNA, he said that chromatin fibers had repeated thickenings. He did not know what it was, but he said that there were repeated thickenings that were found on the chromatin fiber. Later, Audet, a person called Audet, he named these thickenings as the nucleosome. So this is how it was named as nucleosome. Initially they just found that chromatin fiber la vandu thickening irunduchu, and that thickening was named as the nucleosomes. So they found that if this was the simply I am just referring here to the double stranded DNA, a double stranded DNA, idu vandu Woodcock, he said that they had thickenings like this. So it was having repeated thickenings at equal intervals. So this is how, this is what, so this one we call as a DNA, this one we call it as a chromatin fiber. So this is what he said, the repeated thickenings that were found on the DNA molecule, and Audet named these thickenings as nucleosomes. And later when nucleosomes were studied, they found that these nucleosomes contain two important structures. So what is this nucleosome made of? Nucleosome is made of, first one, it is called as a Histone Octamer, and the DNA was wrapped around the histone octamer. So this is what is a nucleosome. So first we will see what is a histone octamer, then we will see how the DNA is wrapped around to make a nucleosome structure. So first one, the histone. So these are actually proteins. So histone proteins, they are actually basic proteins, or they are going to be positively charged proteins, and they are of mainly of four types of histone proteins. And one more thing we have to remember, they are positively charged because of presence of basic amino acids. The proteins they are made of amino acids, so based on only the charge of amino acids, proteins would be charged. So these positively charged histone proteins, they were made of basic amino acids like lysine and arginine in large number. So because lysine and arginine was present in large number in this histone protein, they gave a positive charge to the histone proteins. And the four types of histone proteins are H1, H2, H3 and H4 proteins. So this H2 protein is again of two subtypes. One is H2A and H2B. So these two are the subtypes of H2. So four main types, and these two are the subtypes of H2. And of these four proteins, that is out of H1, H2, H3 and H4, what happened was, for a nucleosome formation, H2A, H2B, H3 and H4 proteins formed an octamer. So H1 was not there in the octamer, only H2A, H2B, H3 and H4 proteins together formed an octamer. So an octamer means, generally the general meaning itself, octamer means it should have 8 units inside it. That is why we call it as an octo. Octo means 8. So it should have 8 units. So how come 8 units were present was, 2 copies of H2A, H2B, H3 and H4 together make up the 8 units of an octamer. So all these join together to form the octamer complex. So we'll just consider these are simple molecules. So this is H2A. So H2A is going to be 2 in number. I am just representing மேலே போடுற ஒரு லைன் வந்துட்டு it is representing that there is one more copy of H2A and H2B and second copy of H2B, H3 and the second copy of H3 and H4 and the second copy of H4. So all together they are going to make, so this structure is what we call it as an octamer. So there are going to be 8 units in it, 2 copies of each of these histone proteins together make up the octamer. So this octamer, so நம்ம இந்த octamer வந்துட்டு instead of drawing like this separately, we are just going to represent it with a single circle. Means it is going to have the 8. So this structure is called as the octamer. So நம்ம வந்துட்டு இங்க 8 subunits உள்ள இருக்கு, together making up the histone octamer. On this octamer, the DNA is wrapped on it, on the octamer. When it is wrapped means DNA is a thread like structure, so it is going to come wind around this octamer. So if this is the DNA molecule, so we'll just consider it like a thread. So this is a DNA molecule, it takes one turn like this and it circles around. So this is what is wrapping of the DNA molecule on the octamer. So this is going to be the DNA that is going to be wound on the octamer. So now DNA வந்துட்டு இப்போ wrap ஆயிருக்குது இல்லையா, that is it is going to make circles. It is actually going to make 1/3/4 turn around the octamer. That is, இப்போ ஒரு circular structure இருக்குது, அது மேலே DNA வந்துட்டு it takes one complete turn and then 1/3/4 turn. It does not take a complete turn. நம்ம இங்கே டயக்ராமில் பார்க்குறக்கு வந்துட்டு இந்த மாதிரி இருக்குது. So we'll just consider, I am drawing it in a different way so that you understand it. So if this is the octamer, so this is the octamer, so this is a DNA, it takes one turn like this and it is going to take another 3/4 turn and then it is going to move out. So it is going to take one full turn, then second round is turn is going to be only 3/4 and then it is going to move come out of the histone octamer, and then is going to be the next octamer here. On the next octamer it is going to take one full turn and the second turn, and it is going to make 1/3/4 turn and then is going to come out, and then is going to be the next octamer here. So the DNA keeps winding like this. So it keeps on moving like So it keeps moving like this. So when this is stretched out straightly, it would look like it is having strings, beads on a string. So this is going to be the beads, and this is going to be a lengthy string on which the beads are attached. So this structure is what we call it as beads on string structure. So this is how an octamer is formed. So an octamer formation and a nucleosome formation. Idhila there is a part, so this is, so I am just marking it here, this is going to be the octamer, and the DNA that circles on the octamer, so this part of DNA that is circling this on the 1/3/4 turn edukudhu illaya, that DNA is called as the core DNA. And இந்த கோர் டிஎன்ஏவும் இதோட இந்த ஆக்டோமரோட கோர் டிஎன்ஏவுக்கும் நடுவில் ஒரு சின்ன ஸ்ட்ரெச் ஆஃப் டிஎன்ஏ விச் இஸ் நாட் அரவுண்ட் தி ஆக்டோமர், ஒரு சின்ன ஸ்ட்ரெச் ஆஃப் டிஎன்ஏ இருக்கு இல்லையா, இந்த ஸ்ட்ரெச் ஆஃப் டிஎன்ஏ, திஸ் ஸ்ட்ரெச் ஆஃப் டிஎன்ஏ இஸ் கால்ட் ஆஸ் அ லிங்கர் டிஎன்ஏ. ஸோ an octamer plus core DNA plus a linker DNA, a single linker for one octamer. So this, so when we are starting it from here, this is going to be the octamer, core DNA plus idoda linker. So this part, so from here to here, this one makes one nucleosome. சில புக்ஸில் வந்துட்டு when you are studying it, நியூக்ளியோசோம் வந்துட்டு இட் கன்சிஸ்ட் ஆஃப் ஒன்லி தி ஆக்டோமர் அண்ட் கோர் டிஎன்ஏன்னு மட்டும் கொடுத்துருப்பாங்க, லிங்கர் டிஎன்ஏவை வந்துட்டு தனியாக கொடுத்துருப்பாங்க. பட் யூஷுவலாக வந்துட்டு நியூக்ளியோசோம்னு நம்ம கன்சிடர் பண்ணுறது வந்துட்டு an octamer, core DNA and a single linker molecule. So this octamer, அதோட core, அதோட single linker. So from here to here, this is going to be one nucleosome. So the histone octamer is going to be made of H2A, H2B, H3 and H4 into 2 molecules, and core DNA and linker DNA. The length of core DNA is going to be 146 base pairs. So around the octamer there are going to be 146 base pairs, and the linker DNA varies from a length of 60 to 80 base pairs. And normally for a nucleosome it is considered there are going to be 200 base pairs. இது ஒரு approximate number தான், நம்ம exact number சொல்கிறது கிடையாது. because 60 to 80ங்கிற number வந்துட்டு வேரி ஆகும். it differs for one DNA to the other, one organism to the other, அந்த மாதிரி டிஃபர் ஆகிறதுனால வந்துட்டு approximately ஒரு நியூக்ளியோசோமில் 200 base pairs அப்படின்னு நம்ம கன்சிடர் பண்ணிக்கிறோம். So this is how the DNA is getting packed in the nucleus of the cell in the form of a bead on a string structure. So this is in chromatin fiber. So bead on string structure. So chromatin or chromatin fiber is called as bead on string structure, and this chromatin or the chromatin fiber during cell division. So நம்ம செல் டிவிஷன் நம்ம படிச்சிருக்கிறோம். செல் டிவிஷனில் வந்துட்டு மைட்டோசிஸாக இருந்தாலும் மியோசிஸாக இருந்தாலும் they are going to get a structure called chromosome. Chromosome formation is going to happen. So during cell division chromosome formation is going to happen. For this chromosome formation chromatin fiber, so chromatin fiber அப்படினாலே already it is going to be a condensed DNA with nucleosome structures. This chromatin fiber with nucleosomes is going to further condense. So further condensation of the chromatin fibers is going to happen with the help of proteins called non-histone chromosomal proteins, in short called as the NHC proteins. So DNA condensation for chromosome formation requires nucleosomes plus non-histone proteins also. இப்போ நம்ம we have forgot one more thing, that is இந்த நியூக்ளியோசோம் ஃபார்மேஷன் இருக்கு இல்லையா, அதாவது வந்துட்டு இப்படி ஆக்டோமர் இருக்குது, ஆக்டோமர் மேலே டிஎன்ஏ வைண்ட் ஆயிருக்கு. இந்த ஆக்டோமரில் இருக்க போகிறது H2A, H2B, H3, H4. H1 ஒரு நியூக்ளியோசோம் ஹிஸ்டோன் ப்ரோட்டீன் இருக்குது. அந்த H1 ப்ரோட்டீனோட ஃபங்க்ஷன் என்ன அப்படின்னா, இது ஒரு பீட் லைக் ஸ்ட்ரக்சர், அதாவது ஒரு சின்ன மோரோர்லெஸ் ஸ்பெரிக்கல் ஸ்ட்ரக்சர் இந்த ஆக்டோமர். அது மேலே வந்துட்டு இந்த டிஎன்ஏ வந்துட்டு ஒரு த்ரெட் மாதிரி சுற்றி இருக்கும்போது இட் கேன் ஸ்லிப் அண்ட் ஃபால் ஆஃப். ஸோ திஸ் H1 histone proteins என்ன பண்ணும் அப்படின்னா, they are going to act like a clamp. அதாவது so this is going to is a H1 protein. So I am writing it here because I forgot to write. So H1 protein, it is going to act as a clamp that holds DNA with octamer. So DNA and octamer vandu they are held together with the help of H1 proteins. And more important characteristic of these histone protein is H2, H3, H4 proteins are inherited, that is they are taken to the daughter cell that is formed during cell division. They are inherited, and H1 protein is not inherited. Because it is not inherited, H1 protein is not considered to be a part of nucleosome. Because it is not inherited, the new H1 vandittu it is not considered as a part of nucleosome, but they help in the holding of the core DNA and the octamer together. So H1 is going to act as a clamp like structure. So this is packaging of DNA. The next we will see some experiments that were performed to prove that DNA as the genetic material. And the genetic material, so there were many experiments. So far we have studied, நம்ம போன யூனிட்லேயும் படித்தோம். அதாவது Mendel was working with chromosomes, that is he did not directly work, but actually he was meaning that genes that were present on the chromosomes, and Sutton and Bovary, when they were studying about chromosomal theory of inheritance, they were studying about chromosomes. And T.H. Morgan, when he was studying, he studied the chromosomes, the genes that are present in Drosophila. இந்த மாதிரி வந்துட்டு all these scientists, they were all studying about the chromosomes, and they were all clear that chromosomes are present in the nucleus of the cell, but they did not know what is this chromosome made of. So by nearly around 1928, that is the genetic material, by 1928 they wanted to prove the genetic material And what was this genetic material made of? So what is this genetic material? Genetic material nu padichanga, chromosome nu padichanga, but they did not know what was this genetic material made of. And after 1928, the person called a scientist called Frederick Griffith he performed an experiment, and the experiment was named as the transforming principle experiment. So the experiment was named as the transforming principle experiment. He performed an experiment, and he more or less came closer to for the identification of the genetic material, but he was not able to prove that. We'll see what is that experiment, and we'll find why he was not able to prove that also. And Frederick Griffith, he was working with a bacteria, that is Streptococcus pneumoniae. So he was working with this bacteria, and he found that this Streptococcus bacteria had two forms of existence, which he called it as two strains of the Streptococcus pneumoniae. One was called as the rough strain, which he named it as the R strain, and there were three strains, R1, R2 and R3. And similarly, he also found one more strain, that is one more form of existence, which he called it as the smooth strain or the S strain. And S strain was also of three forms, one is S1, S2 and S3. And he was actually working with the R2 strain and S3 strain. So there were three different rough strains and three different smooth strain itself, but Griffith was working only with the R2 and the S3 strains. And what was the difference? Why was the names rough and smooth was given was the rough strain did not have any capsule. That is, capsule is going to be a layer that is found outside the cell wall of the bacteria, and because there was no capsule, their outer surface, looked rough. So that is why it was named as rough strain, whereas smooth strains formed capsule. So capsule, mainly made of mucopolysaccharides, made the surface look smooth. So that is why it was named as the smooth strains. And one more difference that was found with this rough and smooth strain was this rough strain was an avirulent strain. That is, it is non-pathogenic or it cannot cause disease. Whereas the smooth strains, they were highly virulent strains. That is, they were pathogenic and were able to cause disease. And what made Griffith work on these, come to the name of the experiment, transforming principle, எதனால வந்துட்டு அப்படி வெச்சாரு அப்படின்னா, when he was working with this strains, he found that few rough strains transformed suddenly into the smooth strains. So that is why he named it as the transforming principle. He was not able to find what was the transforming principle, but he was sure there was some transforming principle present in the bacteria that was able to convert one type of strain into another type of strain. So his experiment that was done, the experiment performed by Griffith was, in his first experiment, what he did was he took the R2 strain. He took the R2 strain of the bacteria and injected into mice. he injected into mice, and it is well known that R2 strain is an avirulent strain, that is non-pathogenic. The mice was able to survive, so the mice was alive. And in his second experiment, he separately he took S3 strain and he injected it into mice. Now we can easily identify, because S3 is going to be virulent or pathogenic, the mice was dead. And in his third experiment, what he did was he heat killed the S3 strain. That is, this bacteria, the S3 strain of bacteria, was exposed to high heat, high temperature, and when it was exposed to heavy temperature, it was killed also. So that strain was called as the heat killed S3 strain, and he injected it into the mice. So because the bacteria was killed, we can easily deduce that the mice can survive. So the mice survived. And in his fourth experiment, what he did was he took an R2 strain. So R2 strain, we know it is non-pathogenic, it cannot cause disease. Mice will survive even if it is injected. Then he took a heat killed S3 strain. So he mixed these two. He mixed these two and then injected into mice. So now, according to his previous experiments, R2 mice survived. Heat killed S3 strain was when it was injected, mice survived. So according to his experiment, he thought this mice, after the injection, it has to survive. But he found that the mice was dead. So what he concluded was, conclusion of Griffith was, he said, the live R2 strain has somehow transformed the dead S3 strain into a living state. So it has transformed the dead S3 strain into a living state. So based in this conclusion was not convincing for any of the scientists who read this. So because of a non-convincing conclusion given by Griffith, his work was not recognized. So his work was not recognized just because of his conclusion that was given by the scientist. Later, 16 years later, that is in 1944, this experiment was done by Griffith in 1928, and 16 years after his experiment in 1944, 3 scientists, Avery, MacLeod and McCarthy, these 3 scientists together, they proved Griffith's experiment to be correct. So they proved Griffith's experiment was correct. That is, there was a transforming principle, and that was responsible for the conversion of actually the R2 strain into S3 strain. Griffith சொன்னது வந்துட்டு R2 strain வந்துட்டு converted dead strain into a living state அப்படின்னு சொன்னாரு. But these 3 people, they said that actually this S3 strain, the killed S3 strain, converted the live R2 strain into an S strain. So that is how the transformation had happened. And the experiment that was done by these three persons, the first experiment, the first, second and the third experiments, that is R2 strain, these three experiments, out of these three, the first, second and third experiment was done in the similar way. So first, second and third experiments was similar to the experiments of Griffith. And now, while performing the fourth experiment, that is, when they performed the fourth experiment, they made a change. Now, before we go into the Avery, McLeod and McCarthy experiment, there's a mistake in the conclusion. Now, first வந்துட்டு மாற்றி சொல்லிட்டேன். Please note it down. The conclusion of Griffith, the conclusion that was given by Griffith, நான் ஃபஸ்ட்டு சொன்னது மாற்றி சொல்லிட்டேன். Please note பண்ணிக்கங்க. The conclusion was the dead S3 strain. So S3 strain was heat killed and mixed with live R2 strain in the fourth experiment. அதில் வந்துட்டு the dead S3 strain had somehow transformed the live R2 strain into an S strain. So அவர் கொடுத்த conclusion வந்துட்டு R strain வந்துட்டு was converted into S strain. இந்த கன்வர்ஷனுக்கான ரீசன் வந்துட்டு இந்த டெட் S3 strain. இதுதான் வந்துட்டு அவர் கொடுத்த கன்க்ளூஷன். But he was not able to prove what was the thing that was helping in transformation. That is why his work was not recognized. But later on, these three people, they found that his experiment was correct and they proved it also. So please note down the conclusion. நான் ஃபஸ்ட்டு மாற்றி சொல்லியிருந்தேன், நான் சேஞ்ச் பண்ணியிருக்கிறேன். Please note this down. And experiment done by Avery, McLeod and McCarthy. So the first three experiments were similar to what was done by Griffith. The fourth experiment, they did the same thing. That is, they took the R2 strain, mixed it with heat killed S3 strain, and it was injected into mice. And based on Griffith's experiment, we know the mice was dead. So mice was dead. What these three scientists they did was, they took this dead mice and their tissues, the tissues of the dead mice was analyzed. And surprisingly, what they found was this tissues, it had only S3 strains, live S3 strains. There were no R2 strains. So what they concluded, that something from the dead S3 strain had converted, so has, or it was entering into the R2 strain and converted R2 into S3. So this is what they have to find. What was this something? This is what they have to find. So now what they did was, they started repeating this fourth experiment with some changes. So this was done by Griffith, and they did it again, and they analyzed the tissues of the dead mice, and what they did was, they started doing the same experiment with some changes. The changes was, along with R2 strain and the heat killed S3 strain, they mixed some enzymes and then injected into mice. And then they found what was happening to the mice. And these enzymes were based on the major biomolecules that were present inside the cell. That was proteins, DNA and RNA. So enzymes that degrade these substances were used. So we'll see how they did this experiment. So first what they did was they took the heat killed. So it was clear the heat killed S3 was transforming the R2 into S strain. So what they did was they took the heat killed S3 strain and this was treated with first proteases alone. So proteases are going to be protein degrading or digesting enzymes. So it was treated with protease and then it was mixed with R2 strain and then it was injected into mice. It was injected into mice. Now they found the mice was dead. So with this what they concluded was, if the protein is that transforming substance, that is the substance that is transforming the R2 into S, then since we have removed all the proteins but still mice is dead, means R2 has been transformed. So proteins are not the transforming substances. So they concluded proteins were not responsible for the transformation. And in the second experiment, now they took the heat killed S3 strain, treated it with RNAs. This is RNA degrading enzymes, and then mixed with R2 strain and then it was injected into mice. And again they found the mice was dead. And with this they concluded RNA is not responsible for transformation. And in the third experiment, they took the heat killed S3 strain, treated with DNAs. DNAs is going to be DNA degrading enzyme, and it was mixed with R2 after treatment with DNAs, and then it was injected into mice. And now they found the mice survived. This shows that DNA is responsible for transformation of R2 into S3 strain. So what actually they proved was the DNA of heat killed S3 strain entered R2 strain, because it was a live molecule, so it entered into the R2 strain and made R2 to form capsules. So once capsules are formed, the R2 is now transformed to the S3 type of Streptococcus. So thus the transforming principle was proved to be the DNA molecule. So this is what the experiment done by Griffith and later on proved by Avery, MacLeod and McCarthy, that the transforming principle present in living cells is DNA, and this can be because it is directing the transformation process, this DNA can be the genetic material. The next experiment is Hershey and Chase experiment. This is also called blender experiment because there an instrument called blender was used, so it is called as the blender experiment. And this was an experimental proof for DNA as the genetic material, because Griffith, Avery, MacLeod prove பண்ணது வந்துட்டு DNA was a transforming principle தான் prove பண்ணாங்க, டைரெக்டாக வந்து DNA genetic material prove பண்ணல. But Hershey and Chase they proved that DNA is the genetic material, and this experiment was done in 1952. And what Hershey and Chase used here was the experimental materials. The experimental materials used by these two scientists were E. coli, a bacteria, and a bacteriophage, that is virus that infects bacteria, that is called as a bacteriophage. And they used these two, a bacteria and a bacteriophage, to prove that DNA as the genetic material. How did they use this was, for this we have to first know the life cycle of a bacteriophage. So in a life cycle of bacteriophage, the bacteriophage, what it does is, first it attaches to surface of bacteria, and then it is going to inject its genetic material into the bacteria. Then the genetic material that is going to enter inside, so the genetic material that enters bacteria will make the bacteria to produce new virus particles, that is new bacteriophages. So virus particles அப்படினா வந்துட்டு இங்க நம்ம சொல்றது வந்துட்டு bacteriophages. So it is going to govern the complete bacteria to produce new bacteriophages, and then it is the new virus particles or the new bacteriophages that are formed, the new bacteriophages formed lies the bacterial cell and is released out. So this is a simple life cycle of a bacteriophage. So the first step in the life cycle of bacteriophage is, this is called as adsorption, that is attaching to the surface of the bacteria. So this is going to be the bacteria and this is going to be the virus, that is the bacteriophage. The second step, it is going to inject the DNA into the bacterial cell, and the next step, after the DNA enters inside, that DNA is going to integrate with the DNA of the bacterial DNA. இது bacterial DNA நான் இங்க represent பண்ணியிருக்கேன். So viral DNA உள்ள வர்றது வந்துட்டு it binds with the bacterial DNA and new virus particles would be formed from it. So once new virus particles are formed, these new virus particles are going to lyse the cell, that is, it is going to break open the cell and release out of that bacterial cell. So this is a simple life cycle of a bacteriophage. And why bacteriophage was advantageous for Hershey and Chase for studying is bacteriophages life cycle. So bacteriophages, எதனால வந்துட்டு they selected bacteriophage and bacteria was studying is the life cycle is clear, that is it is clearly known. இதுக்கப்புறம் இந்த ஸ்டெப் தான் நடக்கும், இதுக்கப்புறம் இதுதான் நடக்கும், அது அப்படின்னு அதோட லைஃப் சைக்கிள் வந்துட்டு கிளியராக தெரியும். இன்னொன்று வந்துட்டு the bacteriophages are made of only two substances, that is protein and second is a DNA molecule. So this is the two substances that is making up a bacteriophage. So now it has now again ஒரு பெரிய டார்கெட்டாக இருந்தது எது ஜெனட்டிக் மெட்டீரியல் அப்படின்னு கண்டுபிடிக்க வேண்டிய பெரிய டார்கெட்டாக இருந்தது. இப்போ வந்துட்டு அவங்களுக்கு வந்துட்டு ரெண்டு தான், இது ப்ரோட்டீன் வந்துட்டு ஜெனட்டிக் மெட்டீரியலா இல்லை டிஎன்ஏ ஜெனட்டிக் மெட்டீரியலா அப்படின்னு நமக்கு தெரியணும். ஸோ இட் மேட் தம் டு செலக்ட் பிட்வீன் ப்ரோட்டீன் அண்ட் டிஎன்ஏ. தட் இஸ் வை bacteriophage was selected by Hershey and Chase to prove. So in their experiment, what they used was they used radioactive sulfur and radioactive phosphorus. எதனால் இந்த ரெண்டு சப்ஸ்டன்சஸை செலக்ட் பண்ணாங்க? எகெயின் எதனால் வந்துட்டு சல்ஃபர் அண்ட் ஃபாஸ்பரஸ் செலக்ட் பண்ணாங்க அப்படின்னா, சல்ஃபர் இஸ் ஃபவுண்ட் ஒன்லி இன் ப்ரோட்டீன்ஸ் and it was absent in DNA, and similarly phosphorus was found only in DNA and it is absent in proteins. So again it is going to become more specific. So they, by using a bacteria, that is E. coli, a bacteriophage and radioactive sulfur and radioactive phosphorus, they proved that DNA is the genetic material. We'll see how they proved it. So in their experiments, we'll just number it for our convenience as experiment number 1 and experiment number 2. In first experiment they took, so what they used in the first experiment was E. coli, bacteriophage with radioactive sulfur. So it was S35 molecule. So radioactive sulfur, the bacteriophage had radioactivity in their sulfur molecules. In experiment number 2 they took E. coli and the bacteriophage with radioactive phosphorus. Phosphorus is a P32 molecule is radioactivity. So they had bacteriophage which had radioactive sulfur in its protein and a bacteriophage which had radioactive phosphorus in its DNA molecule. So now they have to find whether protein is the genetic material or DNA is the genetic material. So first what they did was they allowed the bacteriophage was allowed to adsorb to E. coli surface. So it is the first step, adsorption. So first step, again it is going to be the same thing, adsorption was allowed. So what happened was, on to the bacteria, so on to the bacterial cell, the to differentiate the radioactivity, what I do is I'll just draw it in a blue color so that radioactivity is seen in the sulfur molecules. So radioactivity is seen in sulfur that is present on the proteins, and it is going to have its DNA inside. So this is going to be the first step, whereas here this is a bacterial cell. So onto this, the bacteriophage with radioactive phosphorus is going to get adsorbed. So radioactivity will be seen in its DNA, not on its protein. So protein, we'll draw it with a pencil itself. So because it is not having a radioactivity, and the DNA that is present inside is going to have radioactivity in its phosphorus molecule. So the radioactivity will be seen only in the DNA molecule. Now the bacterial cell, the second step is the bacteriophage injects the genetic material. So the second step is injection of the genetic material. So after injection, what happens is the genetic material is going to enter inside. Here the because protein is present in radioactivity is seen in protein, it is going to be here, and the genetic material is injected. The third step, what they did was, now they try to separate this bacteriophage from the bacteria. So now the cell was agitated, that is it was mixed well, that is a vigorous shaking, adha vandittu agitation. It was agitated with blender instrument. So when it was agitated, so it is called as, the process is called as blending. So it was agitated with blender instrument. So once agitated with the blender instrument, what happens is the bacteriophage that is adsorbed on the surface will come out of the DNA, that is will come out of the bacterial cell. ஏன்னா மேலே ஒட்டிட்டு தான் இருக்குது, உள்ளே போக கிடையாது இந்த bacteriophage. ஸோ என்ன பண்ணும், அஜிடேஷன் விகரஸ் ஷேக்கிங் பண்ணும்போது வந்துட்டு இந்த bacteriophage வந்துட்டு அதோட சர்ஃபேஸ்லேருந்து விட்டு தனியாக வந்துடும். The next step what they performed was, now this mixture was centrifuged for separation. So the next step is centrifugation. So centrifugation பண்ணும்போது என்ன ஆகும் அப்படின்னா, bacteria, because it is heavier than bacteriophages are simpler molecules, so they are going to, bacteria is going to be heavier. Bacteria settles down during centrifugation in the form of pellet, and bacteriophage, because it is going to be lighter, is going to be present in the supernatant, that is on the that is the liquid that is present on the pellet, you call it as supernatant. The same thing happens here also. So now the conclusion was drawn from this pellet and supernatant. So when they were centrifuged, this one that settles down at the bottom during centrifugation is called as the pellet, and what remains back on the top is called as the supernatant. So now they started examining what is present in the pellet and in the supernatant. So where they found the radioactivity, because இங்கே அதை தான் வந்துட்டு கிளியராக பார்த்து கண்டுபிடிக்க முடியும். So based on the presence of radioactivity, conclusion was drawn. So the conclusion was, bacteria infected with bacteriophage with radioactive sulfur, that is S35. இப்போது இதில் வந்துட்டு the first experiment, radioactivity was seen in supernatant, and the second experiment, here bacteria is infected with bacteriophage with radioactive phosphorus. Here radioactivity was seen in pellet, which means that what is present in the pellet was, so bacteria is present in this one. We know bacteria, because it is heavier, it is going to settle down in the pellet, and bacteriophage is going to be present in the supernatant because it is going to be lighter than the bacteria, so it is going to be present in the supernatant. ஸோ இப்போ ரேடியோ ஆக்டிவிட்டி வந்துட்டு அதாவது ப்ரோட்டீனில் ரேடியோ ஆக்டிவிட்டி இருக்கும்போது ரேடியோ ஆக்டிவிட்டி சூப்பர்நேட்டன்ட்டில் இருக்குது, அதாவது பாக்டீரியோஃபாஜில் இருக்குது, பாக்டீரியாவில் இல்லை. மீன்ஸ் வாட்? பாக்டீரியோஃபாஜ் வந்துட்டு we have allowed injection of genetic material. இப்போது ரேடியோ ஆக்டிவிட்டி வந்துட்டு பாக்டீரியாக்குள்ளே இல்லை அப்படின்னா அதோட ஜெனட்டிக் மெட்டீரியல் ப்ரோட்டீன் கிடையாது. ஏன்னா ப்ரோட்டீனில் தான் ரேடியோ ஆக்டிவிட்டி சல்ஃபர் இருக்குது. இங்கே சல்ஃபர் வந்துட்டு இஸ் ப்ரெசன்ட் இன் த சூப்பர்நேட்டன்ட்டில் இருக்கும்போது அதில் அது it cannot be the genetic material, that is it has already injected the genetic material inside. இப்போது சூப்பர்நேட்டன்ட்டில் ரேடியோ ஆக்டிவிட்டி தெரியுது அப்படின்னா, ஸோ ரேடியோ ஆக்டிவிட்டி வந்துட்டு இங்கே சூப்பர்நேட்டன்ட்டில் இருக்குது. மீன்ஸ் வாட்? ஜெனட்டிக் மெட்டீரியல் நமக்கு தெரியும் அது பாக்டீரியாக்குள்ளே போயிடுச்சுன்னு. அப்போது ரேடியோ ஆக்டிவிட்டி எங்கே இருக்குது அப்படின்னு செக் பண்ணும்போது வெளியில் இருக்குது, அதாவது சூப்பர்நேட்டன்ட்டில் இருக்குது. ஸோ ப்ரோட்டீன் தான் வந்துட்டு ரேடியோ ஆக்டிவ். ஸோ ப்ரோட்டீன் cannot be the genetic material. இதே வந்துட்டு இந்த செகண்ட் எக்ஸ்பெரிமெண்ட்டில் வந்துட்டு பெல்லட்டில் வந்துட்டு ரேடியோ ஆக்டிவிட்டி பார்த்தாங்க. அதாவது ஃபாஸ்பரஸில் ரேடியோ ஆக்டிவிட்டி வந்துட்டு இருக்கும்போது பெல்லட்டில் ரேடியோ ஆக்டிவிட்டி இருந்துச்சு. பெல்லட்டில் என்ன இருந்துச்சுன்னா பாக்டீரியா. ஸோ we know பாக்டீரியாக்குள்ளே வந்துட்டு இந்த பாக்டீரியோஃபாஜோட டிஎன்ஏ ஜெனட்டிக் மெட்டீரியல் இஸ் ஆல்ரெடி இன்ஜெக்டட். ஸோ ஜெனட்டிக் மெட்டீரியல் தான் வந்துட்டு பாக்டீரியாக்குள்ளே இருக்கும். So what is the genetic material is the radioactive substance. So இங்கே radioactivity எதில் இருந்துச்சு அப்படின்னா phosphorusல இருந்துச்சு. So with this they concluded that DNA is the genetic material. So this is how Hershey and Chase proved that DNA as the genetic material, and they also gave some characteristics for a substance to be a genetic material. It has to have some characteristics. First is it should be able to replicate, that is it has to double, it has to duplicate itself. அதனால வந்துட்டு it should be able to replicate, and this replication property was found only for DNA and RNA. So proteins can never be a genetic material. Second, they should be stable both chemically and structurally, and this was found here. DNA was more stable than RNA. It was found, so but comparatively வந்துட்டு DNA was more stable because second கார்பனில் இருக்கிற ஹைட்ராக்சில் குரூப் DNAல கிடையாது. So there is no open functional groups, so they will be more stable than RNA, and they should allow mutations so that variations can happen in the generations leading to evolution. So they should allow mutations. They found RNA was more mutated than DNA because it was in a single stranded form and they had more reactive groups. Adanala mutation was more in case of RNA. And one more is they should be able to express themselves, that is the DNA or RNA have to express themselves in form of the Mendelian characters. Mendelian characters, here we talk about the genes or the factors. So they have to be able to express their genes, and this was possible both for DNA and RNA. RNA can directly undergo translation. Now translation படிக்கும்போது you will understand. Translation to form proteins, whereas DNA required an intermediate RNA stage to form the proteins. So it was found that DNA was more safe and a stable genetic material compared to the RNA. This is how they proved that. Again, it was found that both DNA and RNA can act as genetic material. அதுலேயும் வந்துட்டு DNA was the most stable genetic material அப்படின்னு வந்துட்டு they proved it. So with this video we have completed the experiments for the proof of genetic material. Next video will start with the functioning of DNA molecules, that is replication, transcription and translation. Thank you students.